Good afternoon, everyone. My name is Brian Fregoso, and I'm a senior undergraduate student whom will be representing Dr. Erin Rasmussen's lab and the phenomenal graduate students under her care. Today, I'll be speaking on the findings of our study, which is titled Effects of Visual Cues on Conditioned Salivation in Overweight and Healthy Weighted Women. I would like to first thank the lab for allowing me the opportunity to improve on my speaking skills, as well as allowing me to observe the hardships that a graduate student goes through on a daily basis. You are all an inspiration to me and to those around you. Anyhow, our lab was interested in obesity and the mechanisms behind obesity. And so mechanisms is kind of a broad term, so I'll actually specify further. In this study particularly, we were interested in looking at how cues in the environment might play a role in overeating. To further specify this, we wanted to see how the effects of classical conditioned food cues and how they impact healthy and overweight females regarding acquisition and extinction. Firstly, I'll go over what food cues are. Food cues are the viewing of or odors of food cue or food stimuli, advertisements, or any cues or situations associated with food-related memories. These cues serve as a stimuli that elicit food-related responses and then the subsequent food intake. So a very common food cue would be that of the golden arches, which now you're thinking, yes, that is in fact McDonald's. So, research from both human and animal studies indicate that the presence of food cues can increase food consumption, and so it also demonstrated that some individuals are more likely to eat food in the presence of food cues. And this observation suggests that there are individual differences regarding sensitivity to food cues, and there's an actual name for this phenomena. It's called food cue reactivity, FCR for short. So food cue reactivity is the physiological, behavioral, and subjective responses of an individual to food-related stimuli in the environment. So if I could actually specify about the physiological, behavioral, and subjective. Physiological is the salivation that you see from food cue reactivity. Behavioral is the seeking of food. So that is me calling Domino's and asking for a $5.99 large pizza. And the subjective is that of cravings. So cravings such as me wanting a pineapple pizza or that of me wanting a peanut butter Dorito sandwich. So in food cue reactivity, high food reactivity has been shown to increase the probability of food consumption. So examining the relationships between food cues and food cue responses is an important step to understanding obesity. Importantly, we first needed to examine the extent to which we could condition and extinguish food cues in a laboratory setting. We also wanted to compare the conditioned responses between healthy weight and overweight participants. This was the overall purpose of our study. We had 23 obese females and 26 healthy females participate in our study, in which we recruited from Idaho State University through the use of SONA. To distinguish individuals as either obese or healthy, we use the body mass index, or BMI for short. And the BMI is quantified by the individual's weight in kilograms divided by the square of the height in meters. The BMI, or I guess I should say, a BMI less than 24 is considered by our metrics as healthy. An individual that has a BMI equal to or greater to 24 is considered obese. Participants underwent classical conditioning with a visual cue paired with a chocolate reward. This chocolate reward was that was used was that of a M&M button. So M&Ms are actually called buttons, which is weird. We then use an electromyography or myogram, EMG for short, which is an electrodiagnostic testing apparatus that is used to study nerve and muscle function. We use this apparatus to measure swallowing rates, which is a proxy for salivation. Participants scheduled for 2 sessions. The first session measured the baseline swallowing responses in the presence of an assigned food cue. For instance, for a baseline, participants were instructed to look at the screen that displayed random shapes. An example of this would be that of a blue triangle, which is shown on the poster with the dog. They stared at the shape for 10 minutes, while swallowing rates were measured. In the same session, we also measured acquisition of conditioned food cue reactivity. During acquisition, participants were then instructed to look at a screen in which 6 shapes were randomly displayed one at a time. However, the specific shape that they were previously assigned to during baseline, individuals were then instructed to eat an M&M, which means that every time they saw the shape they were assigned to at baseline, they were to then eat an M&M. And the duration of the acquisition was 20 minutes in length. The second session was the extinction phase. During post-acquisition, which was the first 10 minutes of extinction trial, the participants were required to look at the screen that displayed the different shapes again. However, during the session, when the conditioned food cue appeared, the participants were no longer prompted to eat M&Ms, and this part of the extinction trial assesses the participant's swallowing rate in the presence of the food cue directly after food cue conditioning. During the final block of extinction, which was the last 10 minutes of the extinction trial, we measured the participants' swallowing rates while they were exposed to no longer reinforced food cue. The dependent variables were swallowing responses that were measured using the EMG apparatus, and the independent variables were the weight category of obese or healthy, and the time, which was either baseline, acquisition, or extinction phase. We used a 2x3 mix design with weight categories as between subject variables and time as within subject variables. A 2x3 mix ANOVA was used to analyze the data. The results from our study found that there was a significant main effect for time on conditioned swallowing responses. A pairwise comparisons revealed that baseline and acquisitions were significantly different, but no significant differences were found between acquisition and extinction. Further, there was no main effect for weight or an interaction. In conclusion, food cues have been shown to increase food cue reactivity, which may contribute to weight gain. More research investigating extinction procedures for food cues is needed. We found that our lab established conditioned responses with humans, but more trials may be needed to see extinction effects. Thank you.